because of the chemical structure of the drug, it may have the capacity to it may have the affinity to the drug receptor. If the drug has affinity to the drug receptor, they may be agonist, antagonist, partial agonist, and inverse agonist. Agonist means that we'll deal in next we'll deal it afterwards. Next, agonist, antagonist, partial agonist, and inverse agonist have the affinity, which means that they have the affinity to bind with that receptor. After the binding of the drug with that receptor, the action of the drug on the receptor may be different. Agonists have the maximal action, which means they have the activity of they have the capacity for the change. For example, agonists have the maximal activity. Partial agonist may have the submaximal activity. For inverse agonist does not have the activity, but in reverse way. In the case of antagonists, they do not have the activity, but they have the affinity to bind with that receptor. After the uh, binding of the drug with the receptor happens, and after the intrinsic activity has been done, then the transducer mechanisms. After the binding, what actually happens in that cell is actually the transducer mechanism. Transducer mechanism means depends on different receptors. There are different types of receptors like G protein coupled receptors, intrinsic ion channel receptors, enzymatic receptors, and gene expression regulators. In the case of G protein coupled receptors, which means that there is a protein called G protein on the surface of the membrane. Increase in cyclic AMP. There are three types of G protein coupled receptors: adenyl cyclase receptors, phospholipase receptors, and channel regulation receptors. In the case of adenyl cyclase receptors, adenyl cyclase is an enzyme. So when the drug reacts with the G protein coupled receptors, increase in the cyclic AMP occurs. As a result, phosphorylation occurs, which alters functions of many enzymes, ion channels, carriers, and structural proteins which are involved in the various physiological process in the body. Next, in the case of phospholipase type of G protein coupled receptor, phospholipase is the enzyme which actually hydrolyzes the membrane to generate IP3 and DAG. IP3 means inositol 3 pyrophosphate and DAG means diacylglycerol. So IP3 causes release of CA3 deposits, and DAG enhances protein kinase activation by CAT. So calcium is actually the regulator of contraction, secretion, intracellular movements, metabolism, and membrane function of the cell. In the case of channel regulation, when the drug reacts with the G protein coupled receptor, channel regulation may occur. It may close or open the ionic channels which are specific to Ca2, calcium ions, potassium ions, and sodium ions by hyperpolarization and depolarization. Next, there are other types of receptors like intrinsic ion receptors. These are the receptors which are present on the cell surface that enclose ion selective channels. When the drug acts on these type of receptors, opening or closing of the ion channels may occur. Next type is the enzymatic receptors. Enzymatic receptors means that they are also they are also proteins similar to the G protein and intrinsic ion receptors, but they are enzymatic in nature, which means they have the uh, drug binding site and also the catalytic site. Drug binding site is present in the outer surface of the cell membrane, and the catalytic site is present in the inner surface of the cell membrane. For example, phosphorylation activation of guanyl cyclase similar to the adenyl cyclase in G protein coupled receptors, and other enzymes which alter the function. Activation of guanyl cyclase and other enzymes which alter the functions in the physiological process in the body. Next type of receptors are the gene expression regulators. Gene expression regulator means that when you look at the cell, there is a organelle of cell called nucleus. Nucleus contain a genetic material which are RNA and DNA. DNA has the information in the form of nucleotides. So this information on the DNA is actually transcribed onto the mRNA. Same information is transcribed onto the mRNA. From the mRNA, actually according to the nucleotide sequence, a particular type of protein is produced in the human body. Then this protein actually carry out the functions in the is a functional component of the cell, so it carries different functions. So drug here actually enters the cell and binds to the intracellular receptors present in the cell. This receptor protein, after receiving signals from the bind to the this receptor protein bind to the specific genes, which means specific DNA, and facilitate the expression of gene to create specific RNA to create specific RNA. So this type of facilitation of the expression of the gene creates a particular type of protein which carries different functions. For example, vitamin A and D are produced in the body because of the signals received when the drug reacts with that receptor. For example, intracellular receptors present in the cell. So drug mainly acts through these four types of action: physical action, chemical action, drug action through enzymes, and drug action through receptors. So I think this might give an overview of the pharmacodynamics of the drug. Pharmacodynamics, there are two groups in the pharmacology, like pharmacokinetics and pharmacodynamics of the drug. Pharmacokinetics means what is the body doing to the drug, body action of the body on the drug, and pharmacodynamics means action of the drug on the body. So this video, I think this video is helpful for knowing or for giving an overall view of the pharmacodynamics of the different drugs which are administered.